Hello, I'm Phoebe Lewis, a supplemental instructor for Dr. Byron Quinn's Natural Science Biology 1 course, and today I will be covering Chapter 5 in my series of review videos that will hopefully help you with your final. Chapter 5 discussed the plasma membrane. So first we're going to discuss the components of the plasma membrane. We're going to discuss diffusion, active transport versus passive transport, and a little bit on the sodium-potassium pump. Then we're going to discuss osmosis and how it affects plant cells, particularly in the tonicity. And then we're going to discuss exocytosis and endocytosis. So let's jump in. First, let's talk about the components of the plasma membrane. As you can see here, the plasma membrane is made up of several different things. It is once described as the fluid mosaic model of the plasma membrane because there are so many things that make up this membrane. Well, the main component of the plasma membrane is this phospholipid bilayer, as you can see right here. We'll discuss that further on. Another main component of the plasma membrane are these proteins. These proteins serve several functions. Cholesterol plays an important role in the plasma membrane. It regulates the fluidity, and the glycocalyx, which are just sugar chains, play an important role in recognition. So first, let's talk about this phospholipid bilayer. It's important to understand the structure of a phospholipid. So the phospholipid contains a phosphate group at the head, which creates this hydrophilic, which means water-loving, hydrophilic head, and it has some fatty acid tails, which of course are hydrophobic. And so, in essence, when you have a bilayer, you have two layers of phospholipids. And the layers, the fatty acid tails, are going to point into each other because both of them are hydrophobic, meaning they are water-faring. Now, this provides an important characteristic of the phospholipid bilayer. With the fatty acid tails being hydrophobic, it allows hydrophobic molecules, so molecules that are also hydrophobic, to pass freely through the phospholipid bilayer. However, hydrophilic molecules and larger molecules are not able to pass freely through the phospholipid bilayer. This is an important characteristic. The other component of the plasma membrane that we'll be discussing are the proteins. The proteins serve a variety of functions in the plasma membrane, including structural support. As you can see, this particular protein is anchored to parts of the cytoskeleton, so it provides structural support, recognition, communication, and transport. And we can describe these proteins in two different ways. We can describe them as integral, meaning that they span the plasma membrane. So this particular protein goes all the way through the plasma membrane, as does this protein, as well as this one. But there are also other proteins that only go about halfway through, and we call those peripheral proteins. So this is a peripheral protein right here, and then we have integral proteins. Remember, integral proteins go all the way through the phospholipid bilayer, whereas peripheral proteins only go about halfway through. Diffusion is another important principle that we learned earlier this semester. Diffusion says that molecules are going to move from an area of greater concentration to an area of lower concentration until they are evenly distributed. So when you're going from greater concentration to lower concentration, you're going down the concentration gradient. And all the concentration gradient is just a difference between the concentration of molecules. So this figure right here, let's go back. This figure right here gives a good depiction of diffusion. So when this red dye is dropped in, you have a high concentration of molecules right here in the center. Now, according to diffusion, the molecules are going to go from an area of high concentration to an area of low concentration. So as you see here, that is exactly what's happening. The molecules are beginning to diffuse. So they're, in essence, beginning to spread out. You can see that right here, until they're evenly distributed, as shown in this last part of the figure right here. Cellular transport is also a very important concept that we learned earlier this semester, and diffusion plays an important role in cellular transport. You have two types of transport that we discuss: passive transport that does not require ATP or energy, and active transport that requires ATP or energy. Now, there are two types of passive transport: simple diffusion and facilitated diffusion. First, in simple diffusion is just that, just the concept that we learned. As you can see, out here we have a high concentration of molecules. According to diffusion, we're going to have molecules moving to an area of low concentration until they're evenly distributed. So in simple diffusion, um, molecules can move straight through the phospholipid bilayer and into the cell through the process of simple diffusion. Now, in facilitated diffusion, we require a protein. As you can see, you still have a higher concentration of molecules up here, and they're going to an area of low concentration. So, in the case of facilitated and simple diffusion, they are moving down their concentration gradient. However, in the case of facilitated diffusion, you have either larger molecules or molecules that are hydrophilic and can't get through that hydrophobic tails or inside of the phospholipid bilayer, so it's almost like they require a bridge. And so hydrophilic molecules require a bridge to get over the phospholipid bilayer, and that is what we call facilitated diffusion. Now, active transport is a little bit different. In active transport, you can see we're actually going against the concentration gradient. If you see here, you have a higher concentration of molecules down here and a lower concentration of molecules up here. And so what they're trying to do is they're trying to move these molecules into the cell where there's already a high concentration of molecules. This is comparative to if you have an elevator, and the elevator is full of people, and then it stops at another floor. The elevator opens and some more people try to get onto an already full elevator, which you can imagine that it's going to require energy to squeeze all those people together. So when you're going against the concentration gradient, so an area of low concentration to an area of high concentration, you're going to need energy, and that is called ATP, and this is what we consider active transport. One type of active transport that we vaguely discussed earlier on in the semester is the sodium-potassium pump. The sodium-potassium pump uses ATP to pump two important ions, sodium ions and potassium ions, in and out of the cell. So, sodium-potassium pump is a type of active transport. Osmosis is another important principle that you all learned earlier in the semester. Osmosis is the net movement of water from an area of low solute concentration to an area of high solute concentration. So let's refer to this figure right here. As you can see, the solute is of course the salt, the solvent is the water. Well, what you're going to end up seeing in osmosis is not the molecules that are moving. It's actually the water that's moving. And so how I think about it and what helps me remember osmosis is that the solute, or the salt in this case, kind of acts as a magnet towards the water. So wherever there's a high concentration of solute, it's going to pull the water towards it. So as you can see, there's relatively equal levels of water in both of these jars. Well, when you pour the solute in there and through a semi-permeable membrane, what you can see here is that the water is actually going over to this side. So the water level has decreased over here and has increased over here, and that is the principle of osmosis. Now, osmosis can affect the plant cell, and we call this tonicity. So we have three main circumstances that we talk about when we talk about osmosis, tonicity, and animal and plant cells. Hypertonic refers to when there is a higher solute concentration outside of the cell than inside of the cell. So let's think about that. Higher solute concentration outside of the cell than inside of the cell. Well, one important thing to remember is that one major component of both animal and plant cells is water. So if there's a high solute concentration outside of the cell, you have to remember that that water is going to be drawn towards the solute. So in the case of the animal cell, the water is going to be leaving the animal cell, which is going to cause it to shrivel up. Same with the plant cell, and that can cause wilting in your plants. Isotonic surroundings occur when there's a relatively equal amount of solute inside and out of the cell. So you have an equal coming and going of a balance, basically, of solute concentration, which is extremely healthy for the animal cell, and it provides for healthy surroundings for the plant cell as well. Now, hypotonic surroundings occur when there is more solute concentration inside of the cell than outside of the cell. Remember, as I mentioned, the solute acts as a magnet towards the water. So, if there's a high solute concentration inside of the cell, more water from the outside and other cells are going to go towards that cell that has a high solute concentration. Now, in the animal cell, this can be very dangerous. This can cause your cells to rupture or burst because they're filled up with water. Now, in plants, however, you have that sturdy, rigid cell wall. So when the water coming in from around the cell is oops is going into the cell, it's actually going to make it extremely turgid, and it could be a healthy state for the plant cell. Another principle that we talked about was exocytosis. Exocytosis is how plants get, how cells are able to get things out of the cell. And so they form the transport vesicles, mainly from the Golgi apparatus, that fuse with the plasma membrane and then eventually get it out. So in the case of protein production, you all remember that DNA transcribes itself to what? mRNA. The mRNA it exits the nucleus and goes to where? The ribosome. The ribosome reads the instructions on the mRNA and translates that into what? A protein. Well, now that we've made the protein, we need to get the protein out of the cell and perhaps to other areas of the body where it's needed. So the Golgi apparatus is able to form a transport vesicle around that protein that we made and fuse with the plasma membrane to get that protein out of the cell and perhaps other places in the body where it's needed. That is the principle of exocytosis, so getting things out of the cell. Endocytosis is how we get things into the cell. Now, the cell may need to get molecules in, so how we're able to do this is two main ways: phagocytosis and pinocytosis. Dr. Quinn teaches phagocytosis is mainly what we call cell eating. So it basically just, again, forms a vesicle around whatever it's interested in and draws it towards its plasma membrane where it basically brings it into the cell. Pinocytosis works the same way. It's what we call cell drinking, and it forms vesicles around the molecules or water molecules or whatever it may be, and again fuses that with the plasma membrane to get that into the cell. And so that basically covers chapter 5.